Light microscope studies from the early 1930s distinguished two types of chromatin in the nuclei of non-dividing eukaryotic cells: a densely staining and highly condensed form, called heterochromatin, and a less condensed form, called euchromatin. Heterochromatin was originally defined as the portion of chromosomes seen to be highly condensed during interphase. Insight into the nature of heterochromatin was first gained through studies using X-rays to induce random mutations in Drosophila back in the early 1930s. The high-energy X-rays cause DNA damage that can disrupt genes and alter the appearance of the progeny of mutated flies. For example, one of the first mutants isolated this way was caused by the disruption of a gene involved in eye pigmentation that caused the normally red eyes of flies to appear entirely white. The gene that was disrupted is called the white gene. Another mutation affecting eye color that was isolated in similar experiments caused a variegated appearance, meaning some areas of the fly eye were the normal red color and others were white. Upon further analysis, this eye color variegation was found to be due to a change in the normal chromosomal location of the white gene. The white gene is normally located within euchromatin and is expressed, leading to normal eye pigmentation. The mutation that leads to variegated eye color, however, is the result of a chromosomal rearrangement called an inversion, which altered the normal location of the white gene to be adjacent to a region of heterochromatin shown here in green. These observations were the first to suggest that the locations of genes within chromosomes was important for normal gene expression, and that heterochromatin was somehow able to spread into the white gene at its new chromosomal position and inactivate or silence it in some cells and not others. This led to the term position effect variegation. Barrier regions normally flank heterochromatin and prevent spreading into euchromatic regions. In the variegated mutant, however, the barrier region no longer flanks the heterochromatin, which can now spread into the euchromatic region containing the white gene and inactivate its expression. In the developing eye, silencing of the white gene is variable and doesn't occur in all cells. However, when silencing does occur, it can be inherited through cell division. This results in sectors of cells that lack red pigment, resulting in the mottled variegated appearance. Decades after the initial observations of variegated mutants in Drosophila, clever genetic experiments were designed to reveal the molecular nature of position effect variegation. These studies involved further X-ray mutagenesis of the variegating strain to identify new mutations that either suppressed the position effect, leading to a more normal-looking eye, or enhanced the variegating effect on eye color, leading to even more reduced expression of the white gene. These mutations were called SUVAR mutations for suppressors of variegation, and EVAR mutations for enhancers of variegation. Tests were then performed to identify the nature of SUVAR and EVAR mutations. Interestingly, some of these mutations were found to have occurred in genes that encode factors involved in the regulation of chromatin. The most basic level of chromatin organization consists of 147 base pairs of DNA, shown in red, wrapped almost twice around an octamer of histone proteins, shown in yellow, in a structure called the nucleosome. The unstructured tails of core histones stick out of the nucleosome and can be acted on by various proteins to generate a wide variety of covalent histone modifications. Since SUVAR mutations suppressed the ability of heterochromatin to spread into the white gene, these mutations were likely to have disrupted genes involved in either the assembly or spreading of heterochromatin. One of the SUVAR mutations, SUVAR39, was found to inactivate a gene that encodes an enzyme responsible for methylating a lysine located at position 9 on the tail of histone H3. We now know that lysine 9-methylated histone H3 is a histone modification typically found in heterochromatic regions of the genome. This histone modification can recruit a protein called heterochromatin protein 1, abbreviated as HP1. In addition to binding to chromatin modified in this way, HP1 has the ability to recruit SUVAR39, the lysine 9 histone methyltransferase, which can then act to methylate the histone H3 tails of adjacent nucleosomes. This creates a histone reader-writer mechanism, whereby the repeated recruitment of additional HP1 and lysine 9 histone methyltransferase results in the spreading of lysine 9 methylation along a chromosome. HP1 also has the ability to bind to itself, causing the modified nucleosomes to form a more condensed chromatin state that can silence the expression of genes. In this way, the spreading of heterochromatin can alter the expression of nearby genes whose DNA sequences remain unchanged. The study of heritable changes in gene expression that are not caused by changes in DNA sequence is called epigenetics. The selective packaging, or unpackaging, of a region of DNA in heterochromatin is one of several different mechanisms that cells can use to control genes epigenetically. Such epigenetic changes in gene expression help to explain how different cell types of a multicellular organism can all contain the same genetic information, yet display vastly different functions. For example, while liver cells and cardiac muscle cells both have the same genetic information, the expression of this information is different, which leads to them having very different cellular functions. Understanding the molecular mechanisms involved in epigenetic phenomena is also important for treating disease, since an abnormal alteration of histone modifications, and therefore gene expression, has been found to be involved in diseases such as cancer. The study of position effect variegation and the identification of genes involved in the regulation of gene expression is just one of the many examples of how genetic analyses in model organisms are helping us to understand the molecular nature of human disease.